everyone. So first of all, thank you very much for inviting me, and it's really great being here. This is the second time around. Again, so thank you very much, and thank you most. I think most impressive is the meeting with the parents and the caregivers and the children. It's just incredible. It's always very touching, and I brought a student here to just kind of get immersed into this environment, and you feel we get this motivation because us who are a little bit distant from clinicians and the children, it's a little bit, we do it because it's kind of our job. And so when you actually see the parents and the kids, it's very moving, it's motivating. And so thank you very much. Actually, we get a lot from it. So without further ado, thank you very much for staying for the last session. My name is Shai, I'm from Israel, and today I actually changed the title. So what you see in the PDF in the title, and we call it towards a unified genetic therapy for GRIN 2B, associated encephalopathies, and I have to really thank all this work was only being able because of 2B Cured Foundation's support. So it's incredibly important to actually mention the support. Otherwise, we would not be been able to actually be here and show you what we're doing. So I have two aims in this talk. It's to really show you where to what point we've gotten in our research. Again, we don't have a cure. We don't have anything to actually give you, but our intention is to give you as much information about what we're doing, and please grill us as much as possible. Ask us the hardest questions so we can get better. We're doing with the wonderful work of Tali, and of course from the support of 2B Cured, they're really kind of focusing us because, as researchers, we like to kind of, dream and go about, but so they really focus us because we want to take the shortest route between the 2 points, which is from where we are today to actually something that is actually can be given to someone. Anyhow, so we've been working on GRIN 2B variants for the past couple of years. This is a really big a summary of 2 papers that we published. The only thing to take away from this is that we looked at the most severe loss of function GRIN 2B variants that were reported to date, and we decided to focus on this really serious variants. In a nutshell, what they do is whether you have one subunit or 2 subunits or mixed and matched with other subunits, this variant really causes a dramatic drop in glutamate affinity, which means the receptor is basically non-responding to transmission at the synapse, meaning it's a dead receptor, should you want to call it that way. And so this really motivates us to understand what are we going to do with it. And so, as you know, GRIN in large has so many different symptoms with overlapping symptoms. GRIN 2B in particular, because of its embryonic emergence, is really devastating, as I really don't need to mention it to you guys especially. So I just brought from either 2B Cured website and from some really famous papers by colleagues here. As you can see, really intellectual disability and so on, even epilepsy, seizures. And so GRIN 2B variants are so frequent. Depends on the cohort, depends on the country, depends on the size and so on. But we have between 30 to 50% occurrence of GRIN 2B variants among the GRIN patients. They're very heterogeneous. We have mutations spanning the entire gene. It's the longest gene among all GRIN genes, and functionally divergent. So you've heard again and again, so we have loss of function, gain of function, so many different parameters, whether it's glutamate and glycine and surface expression. It's a mouthful just to explain. It's so complex, so many features. It's really hard to make sense of all of this. And so we keep on thinking, how can we treat it? So iPSCs, and we can test different drugs and so on, and we can personalize the medicine. My lab was kind of focused on, we took one step back and we said, how can we kind of find a unified strategy to treat all of these myriad of features and phenotypes and diversity? And so we were a little bit optimistic, I would say. And so we said, can we develop a unified therapy? So, Lord of the Rings, one ring to rule them all. But so we're trying to find one strategy to fix everything. Of course, don't take me seriously. We're not going to fix anything. But we wanted one strategy to be able to be applicable or suitable for many different types of green to be related diseases. And so, you know, we look through the literature, and I just give a handful of papers all the way, you know, dating back from 1999, Joe Chen's lab, where they made the super rats or super mice, and we also have more recent papers from Hogue, our own Amy Ramsey, and so on. And so we thought, what would happen if we just take the good gene, the entire gene, and just put it back into the cell, let the cell do whatever it wants to do, right? So we have most of the kids that we're dealing with, if not all of them, are heterozygotes, meaning they have one good allele, one bad allele. Sorry for the, you know, the simplicity of the way I'm explaining it. And so we said, what if we just introduce a third allele, but a good one, a correct one? And this premise actually, so we call it supplementation, you can call it overexpression, should you want, but we put a third allele. And the idea is that it comes back to a little bit of what colleagues have shown here, especially Ladislav, where the cell neurons in particular, that is a huge kind of endogenous pool of the subunits, and they know, they know how to kind of control what goes up, what goes down. So you have a huge pool of subunits, whether it's 1, 2A, 2B, 2C, and so on. And then somehow miraculously, which by mechanisms that we don't fully understand, they actually go up. And so we were really inspired by a lot of different reports, as well as our own, where if I just overexpress a subunit, you would expect it to kill the cells. Actually, it does not. And I can give you so many different examples. Most of you actually here that do all the biophysics of these tests, what we typically do, we take neurons, wild type background, and we add stuff to it, whether it's the variant or we overexpress the wild type subunit. Most cases, the neurons are okay. Well, functionally, they're not, but they don't die very quickly. It's not a neurodegenerative disease. So in a nutshell, we said supplementation is going to be good for haploinsufficiency, meaning for mutations such as early stop codon inversions, splicing, whatever, meaning we're missing 50% of the subunits, but it's also going to be potentially for gain or loss of function, dominant negative, and that's through a mechanisms that we call dilution. This has been shown again, Roger Nicole has shown this, and so on, where you add so much of a lot of the good one into the pool, and then you increase the probability of a good subunit meeting a good subunit. So you kind of push the bad ones to the side, you dilute them, okay? So as a method of delivery, so we said, great, so we have DNA, we shoot it into the cells, how do we do it? So we chose the AAV, the adeno-associated virus, as our preferable delivery methods for many, many reasons. I do want you to ask me about the recent AAV failure, which is going to be a very interesting thing to discuss. But in very briefly, for those who don't know, it's a very small virus. It's basically a very, very small particle. It's completely proteinaceous. There's nothing inside the virus, so it's just a shell, and you can push stuff in it, genes. These viruses are very stable. They're not teratogenic, meaning they don't incorporate into the chromosomes or into the genome, like lenti and retroviruses and so on. And so they're kind of safe. Excuse me. Highly efficient. You can create in the lab very, very high titers, and you can do whatever you want to do with them. They're naturally neurotropic, so they have a tendency to go towards neurons, which is great for us. And recently, I would say in the last 5 years, I would say, we now have BBB permeable variants, which means it really simplifies the way we address these, the brain, how we reach the brain. So you can inject it through IV or mainly IV, and it just goes through the blood, and then it reaches the brain, and then it does what it does, supposedly. So, but the main problem with these AAVs are they're very, very small. So their advantage is also their disadvantage, because they're limited in what we can put in them. So the GRIN2B gene, as I told you, it's the largest of all genes, and it's right at the top capacity of packaging. And so people come up to me, kind of like Derek, who just said, why haven't anyone done it before? It's because I'm assuming, because I don't know all the data, but we haven't seen anyone package AAV, GRIN 2B in AAV, simply because, I mean, if you look at the numbers, you said, no, it's going to fail. So we came up to be cured, and I said, I was a little bit over-optimistic, and I said, we'll try doing it. Give us some time, we'll do it. Thankfully, they went along with it. And again, there's about 240 clinical trials today using AAVs for a myriad of different diseases, among them about a third, I believe 67 or 70, I can't remember the numbers, just for the brain. And just so you know, there are 3 FDA-approved treatments of AAVs, genetic treatments today, and 3 additional ones actually are now under final approval. So this gave us the motivation to go on. This is the gene, what it looks like. So it's a single-strand virus, DNA virus. So it kind of looks like this in a nutshell. What we did is just add a tag. So we took the entire gene, we couldn't reduce it, minimize it. NMDA receptors, every single mutation that you do obviously can cause something bad. So we took the entire gene, we added a little bit of stuff to it, just tricks that we use in the lab. And we asked, is this going to work? So we just kind of jumped into the water, and the answer is yes. So I'm just showing here a collection of different experiments where we introduced a gene and we infected neurons with the virus, not knowing what to expect, and we label it. We only label the subunit that was overexpressed. We're not seeing, so everything red is what we supplemented to the cell. It's really robust. Usually if you have shitty viruses, you don't get very high infection efficiency, but this one we were very surprised. We did it again and again, and then we tested. It really goes both to the membrane, it expresses inside the cells. We know huge amounts of intracellular pool. Great, so it works. We've done this now, I want to say 50 times. You can obtain very high titers of these viruses. And then we said, okay, this is the largest. If 2B, which is the largest, can be packaged with a little bit of tricks in AAV, can we do this for other GRINs, which are smaller? The answer is yes. It's a resonating yes. And of course, Amy Ramsey can say a lot more about GRIN 1 that has been packaged. There is actually a pending patent that I know of Amy. So we kind of did the same thing with AAV1, with GRIN 1, sorry. We did the same thing with GRIN 2D. We just wanted to make sure, are we imagining it, or is this possible? So the answer is a resounding yes. We can package, you can package NMDA receptors, whatever subunit, which includes AMPA receptors, by the way. They're smaller. So just a description. Very quickly, what we did, we had to make sure that the subunit that we were introducing, because we made small changes that we're not changing. We know a single mutation can cause devastating effects. So we did a lot of different tests. I'm going to go very quickly over it. But the take-home message is that the tagged, HA-tagged GRIN2B subunit, or GLUN2B as we call the protein, is identical as far as we can tell to the wild type. Then the question was, how efficient is the transfection? So if we put a virus, how many of the neurons can be infected? Do we get 5%, 10%, 100%, 20%? So very crude, we grow neurons, huge dishes of neurons in a dish from rats or mice, and then we just put different titers of the virus, and we track them over time, we label, blah, 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 and we get about 75%. This is all automated. AI today does everything. So we get really high transduction efficiency in neurons in a dish. We were very curious what would happen when we overexpress this subunit in just regular cells, just regular neurons that have 2 good copies, and we add a 3rd one. We expected to see huge deficits. Again, in a nutshell, we see no deficits following overexpression of the subunit delivered by the AAV. Again, I'm not going to go over to the details, but if someone wants to, I'm showing it to you, so maybe you can pick up on something. So electrophysiology, as explained before, we don't see any deficits or changes in the neuronal behavior. We ask next, is it lethal, cytotoxic? Is it doing something? Again, in a nutshell, you can see down here, we try various levels of expression. We play with the levels of expression using different promoters, again, genetic tricks. And I can tell you, we see no excitotoxicity. So we don't see dead cells. It's a tunnel assay, excitotoxicity assay. And then we jumped in vivo and we said, okay, that's nice. It's in a dish. What happens in vivo? So we took very young, these are P0, so neonates, very young embryos, or neonates, and we injected directly. So we wanted a lot of virus in the brain, spread, infection, and then we do it in one side of the brain, and then we come back after a couple of weeks and we say, is there any deficits? So phenotypically, the mice behave completely normal, and then we extract the brains, we label, we look, we do electrophysiology, and again, in a nutshell, really, we see no indication of cytotoxicity. And this was very, again, striking. We were relieved to see it. This is a calcium channel. Calcium is important, but it's also very dangerous. Too much calcium leads to, as you know, devastating effects. So we were very happy to see that a mouse that's growing with 3 copies in its neurons of GRIN2B, which is a very highly permeable calcium channel, doesn't do anything. And I'm jumping to the end. We took the viruses, we injected, we didn't even know what doses. We just, injected IV to the tail vein of the mouse. It's very difficult. I hate doing it, but I don't do it. My students do it. So we just inject it and we say, is it going to work? Is it going to reach the brain? Maybe. Is it going to infect the cells? Maybe. Is it going to express the protein? Maybe. What will we be able to detect it? So many maybes. Okay, jumping back, yes, it works. It goes through the BBB, reaches the brain. We cannot predict a priori where it's gonna express the most. We see olfactory cortex, sometimes more hippocampus. It really goes everywhere. We don't direct it. We can't direct it, to be honest. The only thing we can do is kind of control where our DNA is gonna be expressed. Okay, so for the researchers, we do this through promoters, obviously. So reaches the brain, expresses, and again, these mice show no phenotype, no, unilateral deficits because we injected in one side, kind of like the Parkinson mice. Anyhow, that's great. So we have the tool. At least we have the tool. We can package GRIN2B, very important. The next step was, okay, we need a good model. Can we fix some kind of phenotype? So again, with the help of Tali and to be cured, we created a knock-in model based on one of the most severe GRIN2B loss of function. We call it G689C. It's, just remember that it's a heterozygote GRIN2B knock-in. So I'll show you what we do see. In a nutshell, first of all, these animals look completely normal. They reproduce completely normal. They give rise to very normal litter sizes. So we're talking between 8 to 10 neonates. At large, they, just like most of our colleagues here show, these GRIN mice or rats, mice, are kind of normal. They do not really reflect what is going on with the children or diseased adults. But what we do see, they're smaller, a little bit shorter, but what's very striking is their weight. So striking developmental delay. This has been shown again and again by people here, reports on GRIN2D mice and so on. Females start off smaller, but they grow, they gain weight normally as their wild type counterparts. So litter mates, everything has to be we always do siblings. So, heterozygotes versus normals. But the male is very interesting. They actually fail to thrive. So they start out smaller and they kind of, they don't continue in the same trend as the females. Now, we don't know why. But microbiota or as Amy was showing, metabolomics or some additional things are happening here, not just brain diseases. We checked for protein levels. So these mice have one good copy, one knocked-in allele. And again, we see very striking differences between males and females in the brain. So in males, we tend to have slightly higher levels of of GRIN2B at large expressed, I would say the best hypothesis is that, because it's such a very strong deficit, the cells are trying to, to express more to perhaps correct some phenotype. But in the females, we do not see this. And then I was very curious whether we can, so we've never seen homozygote neonates, meaning we never saw mice that were born naturally that have two bad copies. So we can take an inherited homozygote. So I asked my students whether we think, genetically we should have, if we cross two heterozygotes. Long story short, we find embryos that are completely homozygote. I'm sorry. So we find homozygote embryos very early on, but they never get, they do not go through the whole cycle of pregnancy. And it's not because the mothers eat them or cannibalize them. We've looked into it. They literally either something happens during, the physicians can tell us more, what happens to the embryos that do not meet full term. Anyhow, but in these animals, there's about a 6-fold increase in the amount of GRIN2B expressed. It's as if, the cells are trying to express more and more and more and more to try perhaps to correct some deficits. Electrophysiologically, I can tell you that the females, they tend to have a little bit less LTP, which is our indication of capacity to learn. But this also tells us the whole chem kinase and the whole machinery is actually completely normal, intact. LTD, again, we don't see any striking differences between the males and between the wild type and the heterozygotes, whether it's males or females. Naturally, we went to the Morris water maze to test their ability to learn. They learn as well. They're pretty smart as their siblings. I'm going fast because if there's nothing important, interesting, I'm just going fast. But what we see is complete hypoactivity of these mice as opposed to other, I think Vladislav was showing hyperactivity. We see a huge, look at these two graphs. So these are very simple activity cages where you just put the mouse and it just walks and walks and walks. There's a laser beam, and every time it crosses the laser beam, it counts it as 1. So daytime, nighttime, during the day, these mice are, they're nocturnal, so they tend to be less active. And the heterozygotes, so the sick mice, are really hypoactive. They just don't move. And it's not because they have any motor dysfunction. We checked the deficits, we checked for it. And during the night when and you can see the numbers, here are very, very different. At night, they're very active. They're always walking, exploring, whatever. And again, our mice are just hypoactive, completely hypoactive. And we see something very, very similar in the rotorod experiments where, it's like a bar that turns and turns and turns, and these animals have to, walk and learn how to kind of catch up. Not significant, although we've done a very large number of numbers, males, females, hets, and wild type, but we always see this tendency. They're always kind of lazy or hypoactive. But again, they do not have any problem with their motor abilities. We tested this with water maze. If we push them, they will swim. They can do it very well, but they just need a little bit of motivation. These ones are not motivated to move whatsoever. And we see it again in an open field test where the mice kind of move. And again, in the males, we see this very kind of striking. They don't like to move. They just kind of stand there or sit, and they just don't like to move. We don't understand why, although in the females, it's not the same. So we have very clear sex-dependent changes between males and females. There are deficits, both in males and females, developmental delays, definitely. I'm not going to go through it. Electrophysiologically, we said, okay, what's going on? We have to understand more aside LTP and slices and whatever. So we took, neurons, we see no differences, not in hyperexcitability and features of neurons, different currents, and so on. We see no differences. Really, they're identical to their wild-type siblings. So he said, fine, and I'm going to finish with this slide. We did full transcriptomics, so of pups and adult males, females, where we extracted the brains of these mice, and we said, what's different? Is there a compensation mechanism? Something is happening. And to those who are more familiar, look at this. They're identical to their brothers, to their siblings. I've never had, and we've done so many transcriptomic studies, I've never had a study where there was one gene that was different between the different groups. You always get some variability. This is the first time ever I saw this kind of plots where whether it's male pups or adult males or even female adults, really there's no changes in the brain. In the female pups, we do see a little bit more, and we're still kind of trying to dig. And again, we're repeating this whole thing again, just to make sure. And we've also extended to different brain regions or developmental stages, because we really don't understand what's happening. And I will finish with just a general summary. So GRIN2B and other GRINs and GRIA can be efficiently packaged within BBB permeable BBB penetrant AAVs. We can obtain very high transduction efficiency in vitro and in vivo. As far as we tested, we don't see any negative effects, whether it's functionally, electrophysiologically, or staining and other biochemical methods. Now, the correction of phenotype, I know you're all probably very, very eager for me to be able to give you something. I'm very, very cautious. I don't like to kind of jump the gun, but we've injected, I want to say, dozens of animals now. There's different methods of injecting. We've injected dozens of animals that are growing now. We've done iPSCs, we've done organoids with the viruses to see if we can treat something just like Shani was showing with excitability or different electrophysiological deficits, and see, can the virus actually correct it without drugs, of course. So we have two mouse models. I showed you this one, but we're also using the KO rats from the Safari Foundation. It's the one that has it's a heterozygote knockout. I think David Wiley was showing it last year. We also don't see any major phenotype in that. We don't understand why. And I would like to finish with thanking, first of all, this is the view from my office. I'm very proud of it. Although here it's nicer. But so group members really very, very talented. We're a small group, but they're very, very talented and hardworking. To Be Cured really was key to this whole study because of their support. So we were able to do so much in very little time and with very little manpower. Yeah, thank you very much.